Hello, my name is Mark Wappett. I'm doing a PhD by published works at Queen's University Belfast. I also work as a bioinformatician for the drug discovery company Almac, and I'm going to present to you today our web service SynLeGG, which has been developed to aid the discovery of genetic dependency relationships using the large 783 cancer cell line dataset produced by the Broad Institute on the cancer dependency map. The cornerstone of this is transcriptomics data for investigation of these gene dependency relationships, and we've integrated this with functional genomics data from large CRISPR screens and exome sequencing data to establish mutational status in order to propose these candidate gene dependency relationships. So the key relationship that we want to discover here is synthetic lethality, which arises when loss of function events in two or more genes result in cell death, whereas if only one is deleted, the cells remain viable. This could be exploited therapeutically, as in the case of homologous recombination gene mutation, BRCA1, BRCA2 genes, which lead to a dependency on the PARP genes, making these cells sensitive to pharmacological inhibition of PARP using drugs such as olaparib, the PARP inhibitor. So, we're aiming to use this integrated multiomics data set to detect synthetic lethalities and genomic Achilles heels. And so we've developed this algorithm called multisep, which takes the cell line screen and assigns cell line expression to a specific cluster. So between 2 and 5 clusters or expression modes across the screen. And then what we do is we integrate this cluster assignment with either functional genomics data from the CRISPR screen or mutation data from the exome sequencing screen to nominate candidate synthetic lethal gene pairs. And then we also further prioritize gene pairs by adding optional filters. So you can filter for, is it a known gene ontology between the gene pairs? Are they known functional paralogs? Do they share a known protein-protein interaction? And then we have a number of visualization tools that enable you to actually study the proposed relationship between these gene pairs. So how do we know that these results are any good? We've stuffed a load of data into this tool, but do we know that it's actually doing anything useful? Well, the best resource that we could find to benchmark against is a resource called SynLeGG, and this contains a curated record of all known synthetic lethal interactions that have been collated in a single place, and they provide a continuous confidence score based on the evidence to back up these predictions. And so what we've done is we've compared multisep to 2 other algorithms that predict synthetic lethality. Bicep, which was actually the precursor to multisep that we developed, looking at just 2 expression modes or bimodality, and DAISY, which has probably been the gold standard method in the literature for many years for making these kind of predictions. SynLeGG, we used 2 scoring thresholds, so 0.7, which we're calling high, and 0.1, which really includes everything in the database. And multisep outperforms the other 2 methods on predicting synthetic lethality from SynLeGG. In addition, it also finds many more gene pairs than the other 2 methods, and that really, I think, shows the importance of looking at more than 2 clusters, which is what we do with Bicep. We can find up to 5 clusters. We can find that complexity in the data set. So that's the algorithm and the kind of analytics that underpin SynLeGG. And now on to SynLeGG itself. SynLeGG returns a results table. Well, it actually returns 2 results tables, one for the integrated expression and CRISPR data, and one for the integrated expression and mutation data. And SynLeGG, these results are stored under the hood in an optimized SQL-like database. This is optimized for read speed and is indexed as such. The SynLeGG application itself is essentially a Shiny application, but in order to aid scalability, it utilizes Shiny proxy and Docker, instantiating a Docker container for each individual user session. And the controller layer is a series of R scripts which interact with the SQLite database, returning results and or visualizations. And the input to each of those individual R scripts in the controller layer is determined by the user and the actions that they perform in the user interface. So in terms of established synthetic lethalities, we've mentioned BRCA-PARP. We also find the SWI/SNF complex synthetic lethality between SMARCA2 and SMARCA4. And we also bring in protein data, protein expression data here for the cell lines, and we can see that which adds extra validation to the gene pair. So, we've got SMARCA2 mRNA expression binned on the left-hand side, and then we've got protein expression binned on the right-hand side. And finally, novel synthetic lethality. We've prioritized functional paralogs that interact at the protein level, and of these, the most interesting we think is probably RAS binding protein 6A and 6B, which you can see here. And this relationship is most pronounced in lung and pancreatic cancer cell lines. So, in summary, we've provided access to our integrated approach for the wider community to enable analysis and visualization of these genomic relationships, and we welcome any feedback. Thank you.